Yeah, I was in Richmond, and it was on the way driving over. It was not too bad because I was driving kind of late, but all kinds of down trees.
Yeah, it took me an extra hour to get home Thursday because I kept having to. I got within 5 miles of the house, the road's blocked, and then I have to go 20 miles out of the way, and I get within 5 miles, and the road's blocked. Did that 3 times.
Wow.
I was not. I finally ended up stuck like 3 miles from the house, and helping pull a tree out of the middle of the road because that's where it was.
Gotta do what you got to do to keep moving, right?
Yep, and I had about a 20-minute detour coming to work this morning even.
Well, hopefully they'll get all that cleaned up, and I guess hurricane season is pretty close to being finished, but you never know.
Yes.
Well, we'll go ahead and get started. We're moving on to genetics and propagation, and we'll kind of end on the sexual propagation part, and on Wednesday we'll talk more about asexual propagation. So I apologize. For some reason I'm having technical difficulties as well. My PowerPoint is not letting me highlight text. I'm going to try to figure that out and post this presentation with highlights, but just kind of bear with me. You might have to pay a little bit closer attention without the highlights. So let's get to this presentation here. All right. So as we all already know, the plants that we have cultivated and made into crops differ very much so from the wild plants that in many cases are still out there. They're wild relatives we can still find. But the whole process of domestication is what led to our agricultural civilization, and that's not just with plants but animals as well. Fascinating and with the animal part is that some of the animals don't actually even exist in the wild anymore, like the chicken, for instance. It's been so highly domesticated that the wild relative, we're not even sure what it was and if it even exists in the wild anymore. So these differences come from recombination of genes and the DNA really and chromosomes, and then the redistribution of the inheritable traits throughout generations. So let's take a look at this again, and I know we've talked about it kind of in the very beginning, but these are kind of the origins of crops around the world. And I like this graphic because it shows that there's some overlap in many different parts. For instance, you can see that in North America, we've got grapes. We have wild relatives of grape species and strawberries and blueberries. And they also have some of these at other parts of the world too. So it's the combination of genetics from around the country, around the world, that has given rise to the domestication of our food crops. And as I mentioned, they look completely different. For instance, this is the wild relative right here of the cultivated banana. Doesn't look too appetizing, does it? They were a very small fruit, thicker skin. Also, the seeds were much larger, and therefore there was less pulp. You probably wouldn't want to eat that wild banana if you had a choice not to. Eggplant, of course, probably some of you never eat, but again, it's botanically speaking, it's actually a berry. And you can see it here kind of more in its sort of berry-like shape. Of course, we developed the eggplant, one, to remove toxins from it, because in the wild, that plant family, the Solanaceae, actually host to a lot of really poisonous plants. Nightshade, belladonna, these are some of these really poisonous plants that are in that family. So the wild eggplant is actually not really edible. It's actually has some toxins in it, and domestication of it remove some of those toxins. Anybody want to guess what this is? That's a wild carrot right there. And although kind of native to Europe and Eurasia, we have wild carrots that have been introduced here, so we can actually find wild carrots and eat them. And if you do eat them, you'll notice that they have the same fragrance, but they don't really taste like a good old domesticated carrot. You can see here it does have a tap root, but it's kind of forked, and it has some of these fibrous side roots as well. The modern day carrot, of course, is orange or other colors. There's still some white ones out there too, but they've been bred for different color, for larger size, and of course they taste better as well. This thing up here in the upper right corner is what the Mexicans call teosinte, and in its area of origin, this plant still exists. And actually, in their cornfields, they allow it to grow outside of the cornfields because they say that it invigorates the corn crops that they're growing. So I just wanted to point out there with teosinte that although that food there, it would be much more difficult to eat than our modern day sweet corn, for instance. It's really hard, does not have as much caloric content, not nearly as many starches and sugars in it. But it's also fascinating because they say that it invigorates the crop when it grows near it, probably because we bred out some of the traits for hardiness and nutrient uptake, because we started to supply the nutrients, so the plants no longer needed to get them on their own. So it is a trade-off. We domesticated the crops, we made them much better, more edible, but there are some trade-offs there. Finally here, a natural, wild peach was, 1/100th pretty much of the size of our modern day peach. A totally different flavor. Not much edible flesh there really when compared to the rest of the fruit. 36% of it there was the stone. So we make trade-offs when we kind of recombine these genes, when we domesticate them. For the most part though, we made them totally different than their wild ancestors. However, I would also say that the wild ancestors are still extremely important for particular traits and genes that they have that our domesticated crops do not have. For instance, they probably need less nutrients because they're not producing as large of a fruit or large of an edible part. They are also able to compete with weeds a lot better than our domesticated crops. So let's get into some of the science of it here, going all the way down to the chromosome level. Of course, some of our crops consist entirely of living cells, especially our annual crops. When we get into perennial crops, especially trees, they're made up of both living and dead cells. The living cell consists, of course, of the cell wall, the cytoplasm, and then the nucleus, kind of the brain of the cell. And inside of that nucleus is where our chromosomes are in all living things, which is where most of the DNA is stored, although there's some in other places as well. The bulk of the genetic recombination occurs in the nucleus when the cells are dividing. And in terms of the number of chromosomes, it's the same in all vegetative cells of an entire plant species, for the most part. That's at least in the vegetative cells, it's true. When we get to the sex cells, it's different. So we know as our vegetative cells as being 2N is kind of how we talk about it in plant science, but We also say diploid. We have basically two different sets of chromosomes. In the sex cells, however, the number is reduced by half and termed haploid, or 1N for the chromosome number. And so when the cells divide and then recombine, they get full sets of chromosomes, one from each parent. That's why we have 50% of DNA coming from each parent. And it's the same in plants as it is in people. However, the number, the size, and the appearance of chromosomes does vary quite a bit between different plant species. And in some cases, not necessarily just on the species level, but subspecies or different varieties can have different chromosome numbers as well. And today, there's been a lot of science being put into determining DNA and what effects DNA has on species. So now we know at least all of our crop species and most plant species worldwide, we actually know the chromosome numbers of them. And in some cases, we've actually already mapped out the genome. I would say, here in a few decades, they will probably be all of our crop plants and all of our, probably the wild species too, will have genomes mapped. The molecular geneticists are really getting on that. So what are chromosomes? Well, they look like these long thread-like structures, and of course, if any of you have looked at slides under a microscope of them, you've seen them, right? They kind of make like this almost flower-like shape in a way. Within them is DNA and RNA, deoxy and ribonucleic acids, plus the proteins that help to code them. Of course, DNA, we know, has that kind of spiral double helix shape to it. It is an extremely large molecule that can replicate itself and then transmit information to other parts of the cells. Basically, in plants reading DNA as they're being created, that is what codes what the plant will appear to be. And it's also a polymer, a very large molecule made up of many of these repeating units, which are known as nucleotides. And let's just look at that. When we look at DNA itself, the strand is composed of sugar residues linked by, and that's the hexagonal S here, and linked by phosphates on each side, which then holds these hydrogen bonds. And then connects to, basically, or connected by these nucleotide sequences that have these bases. And this is very important to know, that the letters A, T, G, and C, which are these, base pairs, as we call them, adenine, guanine, thymine, and cytosine. They're all held together by hydrogen bonds and attached to the sugars on each strand that create the DNA. So as in our sex cells and our haploid cells, that DNA, during cell division as well as creating those sex cells, the molecule replicates itself by first unraveling and then coming back together with the new sequence, the pairs of bases. DNA is also known as genetic code. And it's actually the RNA, the ribonucleic acid, that controls the cell growth, because it's basically interpreting the DNA and pairing together from the RNA uracil to thymine, or what we know as the base pairs. So the DNA molecule is actually more like a template where this complementary strand of RNA is formed, and then they recombine to pass on genetics. When we're looking at just genes themselves, which the gene is kind of like the smallest unit there. It's a sequence of triplet organic bases inside of a DNA molecule. It is what we know as the ultimate hereditary unit as part of the chromosome. It cannot be seen even with an electron microscope. And it's really fascinating because genes themselves do not necessarily code for one thing alone. In some cases, one gene can code for multiple things, but often genes are kind of linked together, and it's could be several interacting genes or just one gene that determines a trait, or multiple traits in some cases. And that could be the height or the shape of the leaf, flower colors, the fruit size, any of that. It could be something very simple, like in humans, eye color, or in plants, let's say for instance, a pea. Peas have different colors, as we know, especially the snap peas. And it turns out that there are 3 different genes in control of the color of the pod. And they're also in control of particular nodes and what those colors look like. So it's often multiple genes working together. So an individual gene could have a large effect or a small effect on how an organism appears. And some of them act independently, while others have to interact with another gene in order to convey some sort of trait. If genes are on the same chromosome, they are known to be linked, and they can move from one cell generation to the next as a unit. Of course, that linkage is not perfect, and sometimes chromosomes can break and exchange parts. Genes can also be turned on or off due to environmental cues as well. In each vegetative cell on a plant, we have pairs of each individual chromosome. They're what we know as homologous chromosomes. And genes are also termed as alleles to each other, and that's kind of the position of the gene and whether it's turned on or off to actually determine what is actually displayed. So we can look at alleles as being either the same or different, or dominant or recessive. The dominant gene is one that causes certain characteristic to be expressed, whether it is the organism is homozygous or heterozygous. Whereas a recessive gene causes the character it controls to be expressed only if both alleles are recessive. So generally, we're often seeing dominant genes being displayed. For instance, in humans, red hair is a recessive gene. Other colors like blonde or brown hair would be dominant. And two parents, blonde-haired and a brown-haired parent could have a red-haired kid because they both have the recessive red hair gene. And when combined, that recessive trait is able to display itself. When we're just having regular cell division in any part of the plant, it's called mitosis. And that is the kind of non-sexual just replication of genes. The chromosomes split basically down this longitudinal axis, and then they replicate to produce 2 identical chromosomes. And one of each pair goes into that daughter cell. And they have what scientists call an equatorial plate that develops between them to form a new cell wall and then 2 new cells. And in this case, the daughter cell, unless mutated somehow, has the same genotype as the mother cell. Meiosis, on the other hand, is where we have basically sexual reproduction, the sex cells reproducing. And often they call that reduction division. And it occurs in any plants that are able to, undergo sexual reproduction, especially in the flowering plants. So basically, we go back to the whole concept of the flower, and in a perfect flower, we would have both male and female sex cells on different flower parts, of course. The pollen kind of being like the sperm cell, and as you can see here, those chromosomes have been split. And then when the pollen, that haploid, recombines with the ovule, that other haploid cells there, they recombine to create a full chromosome set, and that is then passed on in the seed, which becomes the sexual way of passing on genetic information in plants. In a more simple plants, we have it's slightly different, but because it often occurs over several generations. They actually call this alternation of generations because it goes from a vegetative kind of mitosis to a sexual meiosis reproduction that then eventually develops an embryo and finally a new plant. So when we actually get down to the seed level, the seed coat, which is developed through mitosis from the female parent, covers both the endosperm and the embryo, which has the genetic information from both parents. So it's really interesting to see that in a seed you have both kind of genetics from the parent as well as within the seed genetics from both parents. The manner in which gametes, the sex cells, can segregate and then reunite to form combinations is kind of listed here for both monohybrid and dihybrid crosses. And what we're talking about here is we're looking sort of at the whole concept of the Punnett squares, which basically what we're looking at, Mendel, the father of genetics, kind of created this system of heredity as he viewed it taking place in garden peas. So a monohybrid cross is basically a cross between two plants where the parents are the same except for just one, at least visible characteristic, one difference in a trait. So here we've got a monohybrid cross coming from a tall and a dwarf pea plant. And you can kind of see here the alleles are listed as DD, large D's being tall, and the two little d's being dwarf, because dwarf is a recessive trait. Tallness is a dominant trait. So when these gametes, you take one from each parent, recombine, it displays a tall plant, usually, because of the dominance issue, at least in that first generation. Then let's take, the DD, the resulting, big D, little d, and recombine that. Then you get some difference. You'll get some that are recessive and dwarf, and then some that are tall. And that's due to this whole segregation concept that produces the three different genotypes in these plants. One being homozygous, that is, that it carries both of the same alleles, and that could either be dominant or recessive alleles, or heterozygous, which means that it contains the two different alleles. The alleles are not the same. Homo being same, hetero being different. And really, heterozygosity is what actually leads to more genetic differentiation. If we just had homozygous parents, then we would not see as much difference in subsequent generations. And here they're kind of looking at a dihybrid cross. Again, we're looking at multiple traits taking place. And again, we would see totally different things. And if you think about this, I mean, usually when we're crossing plants, especially if we're doing like a hybrid between two very different varieties, there are a whole lot of different genes that could be changed and altered as a result of that cross. So a lot of times plant breeders try to look for subtle differences and try to breed just for one trait that is much more easily controlled than multiple traits. Of course, mutations are basically just an error in the replication of genetic code, and that changes plant characteristics. And in many cases, it's these mutations that can give rise to a lot of brand new kind of evolutionary features, and mutations that often really cause a species to evolve. Because we have hundreds of thousands of cells dividing constantly, there are bound to be some errors. And of course, those errors could just be naturally caused as just the messenger RNA had a foul, or it could be caused due to some environmental issue, other chemicals from other plants, or radiation tend to induce mutations. Most of the mutations out there, at least initially, are what we know as deleterious mutations. Basically, these will cause some other negative effects in a plant or crop. And this is something that is actually pretty well historically documented in tulip bulbs. Back in, this is the 1200s in Europe, there was this huge tulip craze. And it was kind of funny because people were really fascinated by these mutations, these crazy colors that appeared in the tulips. However, it eventually caused the death of the plant. So they ended up, paying lots of money in some cases. Some cases people traded real estate for tulips, and then their bulbs eventually died because it was a deleterious effect of the mutation. However, some cases they're not deleterious and they aid in developing new cultivars. As I mentioned, we can actually influence mutation rates with chemicals or radiation, and often we actually do that. As I talked about earlier on with the seedless watermelons, we actually chemically altered the chromosome count of a watermelon to get it to be seedless. Hereditary modifications can also be caused, though, in just changes in chromosome number or structure, where sometimes chromosomes break and then exchange segments. And we would see an alteration of genes being expressed in the altered characteristics of the new plants. So when we talk about these chromosomes, often we're talking about ploidy. And polyploidy is just this particular condition where individual plants have more than two sets of homologous chromosomes in their vegetative cells. And that can arise from an additional duplication of the chromosome sets, which is known as autoploidy, or alloploidy, which is a combination of sets from two or more species. And you can see here in this table, they're looking at polyploidy in grains and tobacco as well. And again, same genus, but different species have different chromosome counts, which basically makes it to where they cannot genetically recombine, at least, and still produce viable sex cells. We can actually control certain characteristics in herbaceous plants by affecting genes in the mitochondria and the chloroplasts, not those that are in the sex cells, but those that are in other parts of the plant. These are interesting because they're only contributed by the female parent. Only the female has DNA in the cytoplasm. This actually can be very useful, though, in determining the hereditary relationships among organisms, and is often used for specific types of genetic engineering. Let's talk about the difference in genotype and phenotype. I know we kind of briefly mentioned it before, but genotype is the genetic makeup of the plant, looking at the alleles, the genes, the chromosomes, and the order in which they are situated. However, phenotype, we're looking at the plant's actual appearance, how the genetics are actually expressed in reference to the interactions with their environment. So we have to look at both genotype and phenotype. Often we might see a difference in phenotype and want to try to reproduce that through propagation, but if it's not a difference in genotype, then we can't really replicate it. It's just that the display of that in its environment. For instance, I had a student, and I'm always trying to encourage students to try to look at this. And if you see unique genotypes of plants that you're interested in, see if you can propagate them. There have been a lot of people that have made a lot of money by doing that. If you can recognize, though, and see whether it's genetics or environment, then that might make the difference in making or breaking. I had a student one time that was looking at catnip, and he found this purple catnip, and he thought that was really cool. But it ended up being around this time of the year when nights started to get cooler, and it caused chlorophyll to die, basically, and these anthocyanin pigments in the chloroplast to make it appear purple. So we did a test on that to see whether it was the environment or the genotype. And basically what he did was take some cuttings and asexually propagate that catnip. And as it started to grow, it grew out of the purple and back into the green. So it was not the genetics, it was just the environment. Let's talk about plant breeding, one of my favorite topics. Really, the big thing about plant breeding really boils down to selection. Selecting female and male parents with unique desirable traits and then crossing them to produce offspring that ideally has both sets of desirable traits. It doesn't always happen like that, though, unfortunately. With a lot of advancement in science, especially molecular genetics, we've actually really made breeding a lot more efficient. In the past, they were years to develop a new cultivar with desired traits because the plants had to grow to maturity to evaluate the characteristics. But now you can actually just analyze the DNA, and once you know which genes control what, you can see that almost instantly as soon as the genetic recombination occurs. And only the ones that are, have the specific traits continue to be actually grown to maturity. Here's just a depiction of some cross-fertilization techniques used in the breeding process. Here in a carnation dianthus flower, where this person has taken these tweezers and collected the pollen, the pollinated, the staminate part of the plant, the anthers there, and use that to fertilize the pistil manually. Often these have to occur under controlled conditions. And it often requires very meticulous attention to detail. The female has to be kept from receiving unwanted pollen. If we use male fertile plants to produce hybrid seed on self-pollinating plants in some cases, then what we have to do, sorry guys to bring this up, they actually had to remove the male parts, also known as emasculating them before they mature.
Yes.
Of course, you don't have to do that with male sterile plants that produce seeds but not viable pollen. So there are some shortcuts around that. But when I was doing some plant breeding, did an independent study in this when I was doing my undergraduate, working with a breeder breeding peas. And peas and beans, of course, are self-pollinating. So what we'd have to do is open up the flower before it's fully open naturally and remove those male parts and apply the pollen from a mature plant to the female part of the plant. It's the simple genes that are the easiest to breed for. And of course, the more genes the plant has, the more chromosomes the plant has, the more difficult it is to get desirable traits. It is possible to develop a homozygous line from a single self-pollinating plant, though. Usually when a population becomes nearly or completely homozygous, it naturally has reduced vigor. And that just seems to be sort of an evolutionary thing in plants. In some cases, they have inbreeding mechanisms that will cause them to have pollen from another plant, a genetically different plant. So hybridization, of course, is big, often a huge thing in plant breeding. And what we're looking at often is the F1 generation, the resulting combination from two very different yet homozygous parents. In that case, the F1 plants are very uniform because they all have the same genotype because they're homozygous. If we look at corn, often both feed corn and most sweet corns, it's almost all now produced from hybrid seed, which has basically doubled yields in corn crops. However, being F1 hybrids, if you actually save the seed from them and then replant them, the second generation is going to be different. We're going to have different genotypes and phenotypes. That's the difference in hybridized versus open pollinated crops. Open pollinated crops are heterozygous, which basically means that open pollinated crops, you can save the seed, and although they will probably slightly differ because of that heterozygosity in populations, they will be more reliable, have more reliable characteristics to save seed from. A little bit about biotechnology, which basically we're using molecular biology and genetics to try to advance breeding techniques, not just genetic engineering, but also breeding techniques. Improvements have enabled scientists to introduce many plants with foreign DNA, as with the whole thing with the genetically engineered crops. We might see enhanced dietary attributes or resistance to insects, or the big one, resistance to herbicides. Introducing a gene to a species that does not have that gene is called genetic engineering. They got it right here. They kind of, several times in the book, they kind of go in between genetic modification and genetic engineering. But really, genetic engineering is when we're, really doing kind of more of the lab science and introducing foreign genes. Oh, here they say it themselves. Genetically engineered is a more accurate term. Good work. And then they go to say that GMO is created each time sexual reproduction occurs with different trait combinations in offspring. It's genetically modified, right? We're constantly having the genetics modified. However, not all genetically engineered plants are the result of gene transfer. Sometimes they actually alter the genetics that are already there, like with the flavor saver tomato, which is never made it to the grocery stores because it had horrible flavor. Often genetic engineering complements but does not replace traditional breeding techniques. We actually have to have both. You can't do it all in a lab. Genetic engineering really, though, is kind of similar to plant breeding in that it all starts with the identification, the selection. In this case, we're looking on the gene level rather than just the appearance level, which could be multiple genes. So they have different goals often in genetic engineering. They're trying to do something totally different, and you have to have, whether you're doing traditional breeding or genetic engineering, you have to have these goals in mind. One thing they do in biotechnology is insert marker genes, and often these are actually carried in on little snippets of genetic information carried by bacteria into the genome. It's really fascinating, but they use these to mark genes so that you can see how they're basically disseminated through subsequent generations of crops. If you can follow that gene, then you might be able to breed for it better. Some of the steps here of genetic engineering. This flower has a gene that produces disease resistance. So they identify those genes using molecular genetic techniques, and then they use a bacteria to actually clip, as they say, that snippet of genetic information from the chromosome. And then they attach that marker gene to it so that they can actually identify it and see how it is actually introduced into the crop. Then they insert that gene into the bacteria. The bacteria then injects that gene into the nucleus of the plant cell. And then it's got the new DNA. Then they take cells from that transformed plant and they basically undergo a process to test it, first of all. And then if it did actually have the gene that they were looking for, in this case antibiotic resistance, then it tests out, they continue to propagate it, and then they grow these new plants and reproduce them. So at this point in science, and you might hear people complain about some of this, that scientists are playing God, but they're only able to move or transfer existing genes. No scientist has been able to create an entirely new gene, so it's not really playing God, right? They're not creating anything new, they're just moving them around. I'm going to skip over some of this here. Talk about, and this is a little outdated, but as of 2008, 86% of cotton, 80% of corn, and 92% of soybeans were genetically engineered to be resistant to herbicides, or in some cases resistant to insects. And of course, since then, that has only increased. Despite the fact that we have, a lot of anti-science going on, where people are kind of anti-genetically engineered crops. It happens, right? People, for whatever reason, are against that. And I mean, really, I myself, I'm kind of against the whole, resistance to herbicides because of the other effects that it could have. However, despite the fact that there's a lot of public outcry against genetically engineered organisms, they're still out there. In fact, they're huge. I mean, any basic, unless it has an organic label on it, any shall I say, processed food in the grocery store, if it's got corn, soy, or wheat in it, 99% of the time it's going to be genetically engineered. So that makes the complication of labeling. All right, let's talk a little bit about propagation and how we can do that sexually or asexually, sexually through seed or asexually through vegetative methods. And that table just shows some examples of the different propagation techniques and plants that are often propagated from them. Successful propagation transmits the desirable characteristics of the original plant to the progeny. And with that, we can, actually use, if it's asexual, it's a clone. And by cloning it, we've created all kinds of different varieties out there that now, even though they have just the same genetics as the original, same DNA, there are, potentially millions of individuals out there that have this same genetics, which could be cause for concern. I'll talk about that a little bit later on. So let's look at our propagation methods. If a plant reproduces true by seeds, no characteristics, or at least visible characteristics, are changed, then we call that cultivar a line. And the line has to be homozygous. That means that basically it's going to be very similar unless mutated. And most of our big economic crops, our grains, our vegetables, and garden flowers, are composed of these homozygous lines. However, in forest species, it's a little bit different because they're seed propagated. They have a lot higher variability. So in some of our tree crops, we don't see that as much. So here we have inbred and hybrid lines kind of depicted here, which basically we have a male and a female. In one case, they're homozygous. In the other case, they're heterozygous, or homozygous for a different trait. And then they're cross-pollinated by taking pollen from one parent, applying it to the other to get that cross-hybridization. A lot of vegetables especially are heterozygous with some of those dissimilar genes controlling different characteristics. So in that case, we often use the vegetative asexual propagation to clone them, to keep them the same genetically. Often there are lots of cultivars out there that originate from a single plant, or just a single plant part that are maintained, and we call those clones because they have the same genetic material. And we'll talk a little bit more about the asexual part of that later on. There are several kind of vegetative propagation techniques that are quite complicated. More on that later. We're looking at seed propagation, though, the actual sexual reproduction of the plant. We have to be careful of the seed sources to ensure that often they're homozygous in many cases. If a homozygous cultivar cross-pollinates with other cultivars or species, then it's got to be separated or cold to prevent pollen contamination and loss of genetic purity. However, in some cases, that could be a good thing. We want, especially in extensive plant breeding projects, or if we want to see the differences of different environmental factors, we actually want a little bit of that variability. Well, I'm going to stop there because we've got a little ways to go and we're out of time. So let's just end there, and let's see, am I still sharing here?
No, no, we can see you.
Oh, all right, thanks. Well, you guys have a good one. We'll continue on with this propagation and get into the sexual propagation on Wednesday, and be on the lookout. I've got chapter 7 and 8 quizzes coming soon. I think we had chapter 6 was due, so I'm going to make sure that everybody was able to get that in, and if they weren't, I'll extend the time period. All right, thank you all. Have a good one.
Okay. Bye, everybody. See you in a bit for animal science.